Neanderthalensis. Extinct species that are closely related to present-day humans. Genomes. Study of whole sets of genes and their interactions within a species, as well as genome comparisons between species. Bioinformatics. Application of computational methods to the storage and analysis of biological data. Human Genome Project. Began in the 1990s. International collaborative effort to map and sequence the DNA of the entire human genome. Virtually complete. Phrase researchers referred to when all of the human chromosomes were sequenced. Three steps to completion of the human's genome. 1. Linkage mapping. 2. Physical mapping. 3. DNA sequencing. Situ hybridization, FISH. Method in which fluorescently labeled nucleic acid probes were allowed to hybridize to an immobilized array of whole chromosomes. Cytogenetic map. The visual appearance of a chromosome with stained location of the genes by FISH. Linkage map. A genetic map with several thousand genetic markers spaced throughout the chromosome. Physical map. A genetic map which the distances between the markers are expressed, usually by the number of base pairs along the DNA. In 1992, researchers compiled a human linkage map of approx. 5,000 markers. How to make a physical map. One, cutting up DNA chromosome into restriction fragments. 2, determining the order of the fragments by making them overlap and then using probes, automated nucleotide sequencing of the ends to find the overlaps. Yeast artificial chromosome, YSC. Carry up to insertion of 1 million base pairs long. Bacterial artificial chromosome, BAC. Carry up to insertion of 100,000 300,000 base pairs. High throughput. Methods that can analyze biological materials very rapidly and produce enormous volumes of data. Whole genome shotgun approach. Approach developed in 1992 by molecular biologist J. Craig Venter. Approach that skips the linkage and physical mapping stages and jumps right into the sequencing of DNA fragments from randomly cut DNA. Genomics. Company started in 1998 by J. Craig Venter. Company Venter started with the intention of sequencing the entire human genome. 5 years later, 13 after the HGP started, Genomics and their associations jointly announced that the sequencing of the human genome was complete. Sequencing by synthesis. Phrase for the development of newer sequencing techniques that results in massive increases in speed and decreases the cost of sequencing entire genomes. Metagenomics. Greek meta, for beyond, an approach in which DNA from a group of species is collected from an environmental sample and sequence x used in microbial communities, Sargassous and human intestine. National Center for Biotechnology, NCBI. www.ncbi.nlm.ni.gov. Genbank. Databases of sequences for NCBI. Last. Software program on the NCBI website. It allows visitors to compare DNA sequences with every sequence in Genbank, base by base, to look for similar regions. Reverse genetics. An approach to find the function of genes by its phenotype. Gene annotation. A process to find all protein-coding genes and its functions from a DNA sequence. Expressed sequence tags, ESTs, are fragments of mRNA sequences from cDNA. Encyclopedia of DNA Elements, ENCODE. Started in 2003. An approach where researchers intensively focused on 1% of the human genome to learn about all of the functional elements in that sequence. Ended in 2007. What did ENCODE look for? Protein coding genes. Genes for non-coding RNA. Sequences that regulate DNA replications. Gene expression, enhancers and promoters. Chromatin modifications. What did ENCODE discover? They discovered that 90% of the region was transcribed in RNA and less than 2% codes for proteins. Proteomes. Full protein sets of a genome. Proteomics. The study of full protein sets of a genome. Cancer Genome Atlas. A 3-year project that started in 2007 with the intention of finding all of the common mutations in 3 types of cancer by comparing the cancer cells with normal cells. Three types of cancer the Cancer Genome Atlas was looking for. 1. Lung cancer. 2. Ovarian cancer. 3. Glioblastoma of the brain. Human gene microarray chip. A silicon glass chip that is placed in patients with various types of cancer disease. The chip enables researchers to know the gene expressed within the patient to help them find treatment. MB stands for million base pairs. Why do humans have less genes than bacteria? Humans have a lot of non-coding sequences. Junk DNA. Non-coding sequences. Introns. Non-coding sequences. Pseudogenes. Former genes that have accumulated mutations over a long time and can no longer produce functional proteins. Repetitive DNA. Sequences that are present in multiple copies in the genome. Exons. Expressible sequences. Transposable elements. Stretches of DNA that are able to move from one location to another within the genome. Jumping DNA. Transposable elements. Transposition. When a transposable element moves from one site in a cell's DNA to a different target site by a type of recombination process. Barbara McClintock. First observed transposition in Indian corn by the change of color in the kernels during the 1940s and 1950s. Received the Nobel Prize for Pioneer Research in 1983. Transposons. Stretches of DNA that moves within a genome by means of DNA intermediate. Copy and paste, cut and paste method. Needs transposase enzyme. Retrotransposons. Stretches of DNA that moves by RNA from a transcript of retrotransposons DNA. Retro DNA to RNA2. Reverse transcript into DNA to inserts. Needs transcriptase enzyme. Alu elements. Similar to transposable elements. Accounts for approx. 10% of the human genome. 300 nucleotides long. Do not code for protein. Most are transcribed into RNA. Functions are unknown. Line 1 L1. Type of retrotransposons. Accounts for approx. 17% of the human genome. 6,500 base pairs long. Low rate of transposition. L1 blocks progress of RNA polymerase which is needed for transposition. Simple sequence DNA. A DNA sequence that contains many copies of tandemly repeated short sequences. Located on chromosomal telomeres and centromeres, suggesting that DNA plays a role in the structure. Short tandem repeat STR. Makes up approx. 3% of human genome. Simple sequence DNA containing multiple tandemly repeated units of 2 to 5 nucleotides long. Multigene families. Collections of 2 or more identical or very similar genes. Polyploidy. A condition where one or more extra sets of chromosomes are resulted from an accident in meiosis X. African clawed frog. EVO-DEVO. Evolutionary development biology, a field of biology that compares developmental processes of different multicellular organisms to understand how these processes have evolved and how changes can modify existing organismal features or lead to new ones. Homeobox. A 180 nucleotide sequence within homeotic genes and some other developmental genes that is widely conserved in animals.